Hello everyone, welcome to one more show. Today we are going to have a brief look on the mechanism of action of the two vaccines of the COVID-19. So basically there are still three vaccines that are now in the market. The first one is the Pfizer, second one is the Maderna, and the third one is the AstraZeneca or Oxford vaccine. So today we will see the mechanism of action of the Pfizer and Moderna's vaccine because both have same type of mechanism of action and both have used same type of technology to some sort of develop the vaccine. So basically, they have extracted the genome of the SARS-CoV-2, which is basically responsible for the production of the spike protein. Let's suppose this is the coronavirus, and this is the genome of the COVID-19. And suppose this part of the genome is going to make the spike protein of the SARS-CoV-2. Let's suppose that's the genomic material that is responsible for the production of the spike protein. As we know, there are 4 to 5 different type of protein which are present in the SARS-CoV-2, like spike protein, M protein, N protein. So these companies have extracted the gene or the genetic code that is responsible for the production of the spike protein. Because as we know, spike protein is basically the protein that is responsible for the entry of the SARS-CoV-2 into the cell through the ACE2 enzyme. So they have extracted this genomic part and they have also checked that whether they make the spike protein or not. So what the next they have done is they have wrapped up this genomic material into a nanoparticle, lipid nanoparticle. And why they have done this? Because messenger RNA or the genetic code which is responsible for the production of the spike protein cannot enter into the cell easily. So that's why they have wrapped up into this nanolipid particle. This is nothing but is the it is, basically it is a cover of the lipid particles, and they have introduced this genetic code of spike protein into this nanoparticle. So what is the no, what is the composition of the vial of the Moderna and Pfizer? So in their vial of the vaccine, they have water, they have sucrose, and they have FDA approved buffers. Which, as we know, they're responsible for the maintenance of the pH. They have no used preservative, no use of antibiotics, no adjuvants. So what is the mechanism that how immunity develops? So this is the lipid nanoparticle, and this is the genomic material of the spike protein, and this is the antigen presenting cell of our immune system, which may be macrophages, which may be dendritic cell, or which may be B cell. So what basically happens is, suppose this is the nanoparticle and this is the genomic material of the spike protein. And once they have injected this nanoparticle into the, basically they use this deltoid muscle for the insertion of the vaccine. So when this lipid nanoparticle is inserted into the deltoid muscle, this genomic material is basically comes out from that nanoparticle, as here we can see. And this messenger RNA, which is basically comes out from that nanoparticle, is basically is translated by the host cell ribosomes. And as we know, when genomic material is basically translated, it ultimately makes a protein, and the protein that is made by the host ribosome is basically a spike protein. And this spike protein that will come out from that cell, and how it comes from that cell and how immunity develops, we will see in next slide. So this is the again nanoparticle, this is the messenger RNA, or this is the genomic material of the spike protein. And now when the spike proteins are formed, and after that they are basically exposed on the surface of the immune cells. I have already mentioned like macrophages, dendritic cell or T cell or B cell. So when these spike protein are basically presented by these professional antigen presenting cell, after that they some sort of activate the plasma cell or the B cell, which then further make the antibodies, and these antibodies then definitely bind with the spike protein, as we can see. And these antibodies then neutralize the spike protein. And this is the one pathway. The second pathway is that these spike protein also activate the cytotoxic T cells or the CD8 cells, which are basically the immune cells. And when these cytotoxic T cells are activated, they then basically produce some sort of the enzymes, caspases or enzymes and perforins, and then these T cells then kill that particular cell which is being affected by the virus. So they are not basically involved in the immunity. Basically, B cells are involved in the immune formation, immune cell or immunity. So basically, this is the mechanism of action of the Pfizer and Maderna vaccine. And the mechanism of AstraZeneca or Maderna vaccine is little bit difficult because they have not used the nanoparticle. They have used the adenovirus, chimpanzee's adenovirus. So this is the mechanism of action of these two vaccines, Pfizer and Moderna. So at the end, I requested all of you to please like, subscribe, and share. Thank you very much.